Okay, so just to clarify, you have a test the next time I see you, which is Tuesday. Yeah. Tell me what it covers. For sure, cardiovascular, synapses and neurotransmitters, and ANS. Yeah, good. And they kind of go together. I don't know if you noticed that. They do. I wish they were in a different order, but I was trying to stay in the same order as the lab. That's Tuesday. We're going to do this material. This material is for your 4th exam. So let's clarify, because people are asking lots of questions and they forgot, and you slept a lot and now you're stressed out and your brain is only partially functional, like mine. So what are the circumstances under which you can not take your final exam? Be very specific. 92.5 or higher on what? Everything without dropping anything. Yes? So if you have a 92.5% or higher on everything without dropping anything. So can you drop a midterm and then not take the final? Not unless you want to fail. Okay? Because you will. What happens if you are not making a 92.5 and you do not show up for the final? I do not drop the final. I enter that 0 and I drop one of your midterms. Okay? So you have to earn that week off. And I certainly hope that you do, right? But what happens if you are not making a 92.5, show up for the final, and make a worse grade on your final than your lowest midterm? I just dropped the final for you, right? So here's the deal. I have designed it so that you are encouraged all the way till the end to get the highest grade that you can possibly get, but the final cannot hurt you if you have taken all of your midterms. It can only help you. So if you've taken all of your midterms and you go in and take your final, your grade cannot drop, but it can raise. Does that make sense? So I want you to keep working, keep trying, get the best you can out of this class, and don't be discouraged until the end of the semester. And don't go on vacation, decide just in the last week. Because, you're already here, you paid for it, you did everything you could. Maybe you had a dumb day and made a terrible test score. There's an out. Okay, so anybody have any questions? Everybody good? Okay. Oh, the other thing I was talking to Maeve about is that there is going to be a survey coming out about the lab, and you're going to be given that survey during the week that you're doing your POPs presentation. Here's my request to you. Realize that the things that they're doing in lab are trying to improve the quality of the phys lab. A couple years ago, everybody hated every single piece of the lab, and now they like parts of it. But what I would like is for intelligent people who are reflective to really give some feedback on what worked and what didn't. The POPs project was in place of a midterm and a final, and I really think it's a good idea, but you guys were the pilot group. So if you can give some careful, well thought out feedback, we can make it what we hope it can be, right? Not I hate it, I hate everything about it, F you, because the alternative is a midterm and a final, and those people tanked, FYI, in the lab. Because did you remember what concentration of who to what's to add to the snooga locker? You didn't, right? And you don't wanna. And that's what the freaking midterms were like. I hated them. So anyway, that's gonna come out. I'm gonna do my damnedest to try to encourage them to give you bonus points on lab for doing that survey. I'll work on that this week. I'm not the one who's curriculum director for Phys Lab, so I just have to try to pitch it to him and say, these people did a pilot project for you. Let's reward them for being guinea pigs, right? Let's reward them. Even the guinea pig gets a snack, doesn't he? Right? Definitely. You ran the damn maze. Might not have won it. Here's a snack. Okay. I guess that's a wrap. I mixed my metaphors, but it's fine. I feel good about it. Okay. All right. So general information. So we're going to do blood. Blood physiology is not long. It gets a little tricky at the end, but it also isn't it's not super complicated except for one little thing about it. Okay. So the general information, let's just talk about blood in general quickly. So you guys did 2 freaking labs over blood. I'm going to smash those 2 together. Don't think I'm not. I've been trying to push that forever. I'm like, why 3 weeks over cardiovascular and no nervous system labs? Why would you do that? But they don't listen to me. But I'm very persistent. It's like one of my most annoying qualities. Okay, so what is blood like? Well, it is thicker than water. It's 5 times as viscous as water. I don't understand that idiom, though, because why are friends water? Oh, no. So it's thicker than water. It's 5 times as viscous as water. It's sticky because of the proteins and sugars in the blood plasma primarily. And it's basically like interstitial fluid because it's like extracellular fluid plus a whole bunch of cells and some extra clotting proteins in it. It's hotter than your average body temperature. And its pH is very, very narrowly constrained. One of the things as you guys go into pathophysiology and go into clinical situations, you will figure out how minor changes in blood pH can cause massive changes in health. So the pH is pretty tightly constrained. 7.35 to 7.45 is healthy. Go a tiny bit above that, a tiny bit below that, you are going to have all kinds of physiological symptoms. It's isotonic to about 0.9% saline, like normal saline, right? And it makes up about 8% of your total body weight. So that last 10 pounds you're trying to lose, it's blood. Just be nice to yourself. It's totally fine. It's definitely not pie. Okay. How much do males have? On average, males have 5 to 6 liters. On average, females have 4 to 5 liters, just because of difference in body size. It doesn't mean that a really tall woman would have less blood than a really short man. That's just on average. Okay, so you know the functions of blood, so let's go through them quickly. Blood is basically a transport pathway for wherever the stuff was picked up to wherever it's going. Oxygen from the lungs to the cells, carbon dioxide from the cells to the lungs, nutrients, GI tract to cells, waste products to wherever you get rid of them. The primary places you get rid of the waste products are kidneys, liver, and skin. And then hormones, we've met a couple, from the endocrine organ to the target cell. That's what travels through the blood. And then blood is also, because it's circulatory, it's also really, really good at body-wide regulation. It regulates body pH because it's got a buffering system within it. And since it regulates the pH of a circulating fluid, it ends up being able to regulate the pH of the whole body pretty effectively. It regulates ion composition from wherever you have a bunch to wherever you have a little. So if I needed calcium and more calcium in one place, I just take it from the bone, right, and take it to that place. Potassium, same thing. It also regulates the fluid volume of the circulatory system because it has high concentrations of things that suck. High concentrations of things that hold fluid in, primarily sodium and the protein albumin. And it's also a protective fluid. First off, because it has circulating white blood cells and antibodies in it, yeah? But then also, it is the only fluid that you have that has the ability to stop fluid loss. It's the only fluid that clots, okay? And then last but not least, it helps both actively and passively in stabilizing body temperature. It's basically a swamp cooler, right? In which it's just sort of circulating from the warm places to the cool places. In addition, because it's primarily water, like the interstitial fluid is primarily water, forming and breaking hydrogen bonds is a way to release and absorb heat. Yeah? So just because it does that. All right, so let's get into red blood cell physiology. This isn't the hard part. We can go through this relatively quickly. So when we look at red blood cell physiology, and we'll do this again when we get to the respiratory system, which I think is last. Do we do urinary or respiratory next? Urinary next. Good. I hate respiratory. That's why I always do it last. I'm like, do I have to do this? If I run out of time, I'm not going to do it. But I'm not out of time this semester. There are like 3 lectures that I hate in phys, and there are at least 3 that I hate in anatomy too. I'm like, all right, let's just do it, whatever. I feel like I just need a shot before I do it. Especially when students are really bored and it's hard to teach. You're like, why? Why do I have to do this? Can I just give you a worksheet? What? I know, but worksheets are so hard. They're harder than lecturing. I can lecture like that. They're hard for me to do. Okay, so red blood cell physiology. Let's talk about our friend hemoglobin. You've met hemoglobin before. Do you remember anything about it? It is 4 peptide chains, to alpha to beta, with the heme group in the middle, and the heme group is iron containing, okay? And it is what makes your blood red, hemoglobin. So it's what makes your blood red. And so let's talk about its functionality a little bit. I mean, you guys know a lot of this anyway, but before you eject your organelles, do you guys remember that red blood cells no longer have a nucleus or most of their organelles when they're mature? Yeah, that's why they have that sort of donut shape, right? Not a complete donut, but you didn't get the hole all the way out. It's called a biconcave disc. During that process of red blood cell development, before they eject the nucleus and the ribosomes and all of that crap, they make a whole bunch of hemoglobin. So primarily what they are, to oversimplify a bit, is pretty much a bag filled with hemoglobin. And hemoglobin is this red iron-containing pigment. Iron red, you know, like iron rust, iron red. So what does iron do when it gets exposed to oxygen? It oxidizes. What does it do to the color? Turns it red, yeah? What does iron do to oxygen on hemoglobin? Turns it redder, yes? So even though my 13-year-old still will not believe me and asks my husband, who is a philosopher and a programmer, if this is right, I tell him every day. He's brilliant, right? But he ain't this kind of brilliant. So I tell my son that your blood is not blue in your veins. And he goes, Dad, is that right? I'm like, Dad, is that right? Well, no, you're asking him. What the hell does he know? So it's not blue. It is red when it's oxidized. It is less red when it's not oxidized. So why does it look blue? You know, it's just basically there are pigments in your skin. There are pigments in the wall of your vessel. You're seeing all of it go through. I figure the next time I'm just going to cut him to prove it to him. No, I won't. I adore him, and also he's bigger than me. So he still doesn't believe me now. Go away. What is... Okay, so where would blood be red? Blood would be red in systemic arteries and in pulmonary veins. Good. Okay. So how much hemoglobin? 100 mils is like, I don't know, like that much-ish. Males and females have different amounts of hemoglobin. Males tend to have about 13 to 18 grams per 100 mils, and females have about 12 to 16 grams per 100 mils. Males also have more blood, but their blood has greater oxygen carrying capacity than females, and we'll look at why in just a minute. It's kind of interesting. So the function of the red blood cells is primarily gas transport. You guys are in the habit of thinking that red blood cells transport oxygen, and that's true, but red blood cells also transport carbon dioxide. So let's talk about that just briefly. Okay, so this is hemoglobin. If you break it down, this is the heme portion, and this is the globin portion. And globin was named globin because it's a glob-shaped protein, globin, yes? Okay, so what happens is you actually carry oxygen on the heme portion, oxygen gas, O2. You can carry 4 molecules of oxygen gas on 1 molecule of hemoglobin. Does that make sense to you? Okay. You also, though, carry carbon dioxide, but not on the heme portion, so it doesn't outcompete or compete with oxygen at all. It's carried on the globin portion. Does that make sense to you guys? So it's carried on the globin portion. So when oxygen is bound to hemoglobin, it's called oxyhemoglobin, and it's bright red. And so, of course, what's going through pulmonary capillary beds, you tend to pick it up. It becomes oxyhemoglobin, back to the heart. Pump it to systemic capillary beds, it becomes deoxyhemoglobin. Back to the heart, back to the lungs, do the same damn thing again, over and over and over until you die. But while hemoglobin is in systemic capillary beds, it also picks up a friend that sits next to it, but not on the same seat, and that's carbon dioxide. So as you go through systemic capillary beds, the globin portion picks up carbon dioxide. As you go through the pulmonary capillary beds, it drops it off and you breathe it out. Okay, does that make sense? Okay, so carbaminohemoglobin is what it's called when carbon dioxide is bound to it. So we will get to this in the respiratory system, but only about 20% of carbon dioxide is actually transported through the blood as carbon dioxide. The rest of it is transported through the blood as this, the bicarbonate ion. And I think you've seen that equation before, haven't you? Okay, so one of the things that it can do if you force it to get transported as this is that you can control the pH instead of carbon dioxide impacting the pH in a way that you can't predict. If you force it and then hold on to the H+, it can't impact the pH. So what ends up happening is we can carry massive quantities of carbon dioxide with minor impacts on pH. So we'll get to that. Okay, so let's talk about how red blood cells are formed, what you need to do it, how you get rid of them. Where do you make red blood cells? You know this. Bone marrow, be more specific. Red bone marrow, exactly. So we make red blood cells in the red bone marrow. That process is called erythropoiesis. Yeah, erythropoiesis. And it's just sort of a sub-process, I don't know what the hell word I'm looking for, of hematopoiesis. So in order to make erythrocytes in the red bone marrow, let's talk about what you have to have, okay? Nutrients that you have to have and hormones that you have to have in order to do this adequately. So if you're talking about erythropoiesis, the first thing I want you to know is that you're doing it relatively constantly, yeah? On a good day, red blood cells live about 120 days before they're actually broken down in the spleen and the liver and you make more. But you've basically always got sort of continuous turnover of red blood cells. So you are doing continuous turnover. Now, of course, when you're growing, you have to increase your blood volume, so you're going to need more erythropoiesis, right? But so let's talk about someone who is growing or someone who had just given blood. What would you need in order to replace that? Well, you need iron, duh, right? Because iron is, of course, a component of hemoglobin. If you do not have adequate amounts of iron, you either will not have adequate amounts of hemoglobin or you will not have adequate amounts of oxygen binding sites on hemoglobin. Does that make sense? Okay. So now, why do you need to continually replace your iron? Well, because you lose iron. Most forms of iron are water soluble and you'll lose some of it in your urine, some of it in your skin. And if you're female and you're of reproductive age, you're going to lose some of it via your menstrual flow. Let's acknowledge something that sometimes people haven't discussed. When you're menstruating, you're not actually losing blood, you're losing vascular tissue. It is actually tissue with a rich capillary bed within it, and you're losing vascular tissue. And women with a really heavy menstrual flow, your iron is pretty decently high before, and then during and after it can really, really drop. And so some women during certain portions of their menstrual cycle will actually become anemic, right? Especially if they're not keeping track of. You can front load the amount of iron if you're really careful about it. Okay, so iron is lost via the urine, the skin, the menstrual flow. But you also have the capacity to sort of control absorption rates with iron. So your intestine, if your iron is low, will increase absorption rates, right? There's some hormonal mechanisms, there's some other mechanisms. So we've got iron coming in from ingestion and you're going to absorb iron into the body. The GI tract controls rates of absorption based on the body's needs. And then if you ever have excess iron, since it's relatively valuable, we actually store it in the liver. So excess iron is actually stored in your liver. Sources of dietary iron. Where does iron come from primarily in your diet? This isn't a comprehensive list. But meat, yeah? Especially, at least one other Southern girl in here. Liver. Did you eat fried chicken livers as a kid? You didn't? Oh my God, I totally did. So I was super anemic as a kid, and my mom would fry up chicken livers and cut them into little nuggets and leave them on the bottom shelf of the fridge, and I'd just toddle in and pop in some chicken livers, right? So not surprisingly, liver is really high iron because that's where animals store their iron, right? I'm not anemic anymore. I don't know. Maybe it works. I also don't eat liver anymore because I know what the hell these things do, man. I'm like, a little education. That's going to change everything. Okay. Egg yolks, beans, nuts, cereal, and leafy green vegetables. Those are your primary sources of iron. Note to self, although it's not very common, since men actually do not lose iron as regularly, they can actually get a disorder in which they have excessive amounts of iron in their bloodstream. Guess what a fantastic treatment for that is? Giving blood regularly. Yeah, giving blood regularly. Interesting. Women don't tend to get that very often, especially during their reproductive years. You also need folic acid. Folic acid is needed for cell division of all sorts, and red blood cell synthesis is a cell division party, and it's a relatively constant cell division party. So it's needed for DNA synthesis to make thiamine before cell division, and sources of folic acid, green plants, yeast, liver, citrus. Note to self, not necessarily related, but since folic acid is needed for DNA synthesis prior to cell division, folic acid is a really, really important nutrient during pregnancy. Even before you become pregnant, super duper important, and it helps prevent neural tube defects. Spina bifida and hydrocephalus associated with it and all that kind of stuff. So I always just take a minute to say this because if you are a woman who could become pregnant and would retain that pregnancy, you need to make sure you've got adequate amounts of folic acid in your diet because neural tube defects happen in the first 2 weeks of pregnancy. How many of us knew we were pregnant in the first 2 weeks? Hell no, you didn't. Okay, I mean, maybe you did, but I don't know anybody else who did, right? As a matter of fact, you only that would be 3 weeks after sex, you know? I don't know. I sure didn't. Okay. Vitamin B12, also needed for DNA synthesis, dietary sources of vitamin B12. If you are vegan, you have to pay attention to your vitamin B12. I'm not saying being a vegan is unhealthy. I'm saying being a vegan who doesn't know about physiology is very, very potentially unhealthy. So the primary sources of vitamin B12 are from animal products, not necessarily from animal flesh. You can get it from milk and eggs and those kinds of things. But you can also find it in nutritional yeast, but you have to pay attention to your vitamin B12 levels if you're, and some other vegan products, but you have to pay attention to it. Okay, and then a hormone. I know you guys want another hormone, like a hole in the head, but here is one. Epo Erythropoietin is actually kind of a cool hormone. It's a hormone that comes from the kidney. When the kidney detects, so we talked about a hormone last time, and for some reason it took me a while to figure out not to smash these two together. So last time we talked about renin, which was released from the kidney when the kidney detected low what? Blood pressure, okay? And its negative feedback activity was to raise blood pressure by 2 mechanisms. Good? Erythropoietin is also released from the kidney, but when the kidney detects low oxygen, regardless of blood pressure. You with me? And so it's going to target the bone marrow, and since you are a brilliant physiologist who knows about homeostasis and negative feedback, what do you predict it's going to do to the red bone marrow? Increase erythropoiesis, exactly. Okay, it's right there in the name, erythropoietin. It's a peptide. It comes from the kidney and targets the red bone marrow and causes erythropoiesis. Specifically, it targets stem cells called myeloid stem cells that are the precursors to red blood cells and some other things. And the body produces about 2 to 3 million erythrocytes per second. Damn, right? You thought you were lazy. No, you're just busy. I'm just busy doing other stuff, right? Just busy doing homeostasis. It looks like I'm doing nothing. You have no idea. Okay. EPO, not secretion. I don't know what that is. Secretion is triggered by hypoxia detected in the kidney. What could cause hypoxia? Any number of things could cause hypoxia. If you just gave blood, wouldn't it make sense that you'd have less oxygen in your bloodstream? Yeah. So hemorrhage, destruction, something like that. High altitude. So that gets those of us who came from sea level or closer. Anybody from Louisiana? Because you were basically an underwater mammal. My husband's family is Dutch, so they're all underwater aquatic animals anyway. That's not even supposed to exist. So what happens when you come from lower elevation is, of course, there's higher amounts of oxygen available. And then when you're me, you move here, right? And then you can't breathe. And you think it's because of the oppressive culture, but it wasn't. It was because, editorial comment, shut up, Melanie. So it was because there was little oxygen available in the air, and I had relatively low levels of hemoglobin because I was from sea level, right? So high altitude, also COPD, which is chronic obstructive pulmonary disease. That's anything causing problem with air getting in. Or if you start exercising a bunch, right, then your demand for oxygen goes up and it can cause hypoxia. So what that means is, of course, if I start exercising very regularly, and again, I get that hypoxic error message very consistently, what will happen is my kidneys will go, oh, you're going to exercise 3 times a week? More than 1 week? Then it will say, okay, I trust you. It might not, because it's February. It doesn't realize that it shouldn't trust me. And then it's going to release EPO, which actually causes me to increase red blood cell synthesis. Yeah? But it doesn't do that the first time you exercise, because it doesn't trust you. It's met you. Okay, and then testosterone. Testosterone is interesting in a million different ways. Of course, males have more testosterone and other androgens than females do. Testosterone can also stimulate the kidneys to release EPO, and that accounts for the different amount of hemoglobin, yes, and the different amount of red blood cells in males versus females. So if you looked at a cubic millimeter, and a cubic millimeter is like a big drop, right? About a big drop. It's a mil. Then males have about 5.4 million red blood cells per cubic millimeter, and females have about 4.5 million. So this explains the difference between male and female red blood cell count and hemoglobin amount, right? And it also explains something important to understand about males versus females. Males have greater oxygen carrying capacity per unit blood, and they have more blood than females, yes? So let's talk about aerobic activities in males versus females, right? So although none of us in this room, I'm going to say this with ultimate confidence, can beat Serena Williams, right? If Serena Williams had a male cousin that had her genetics and her training, he could probably whoop her. Because, right? Okay. Because the bell curve for aerobic activity in males versus females, they have greater oxygen carrying capacity than females. That's not per individual, right? I don't know, did you guys see that there was like a Twitter battle in which like Serena Williams posted something and some dude, just like random Twitter dude, said, I could take her. I'm like, oh, I'd like to see it. Can we see that? It's like, you can't. Okay. So clinical connections related to red blood cell synthesis. High altitude training and erythrocytosis and blood doping and EPO and all of this kind of stuff. So we've kind of touched on this before. Why do elite athletes train in Park City? Because it's higher altitude, right? And so it's not really a competitive advantage because all of them do it, right? So it's a wash, really. Some of them decide to go the next step, which is blood doping. And blood doping, you can do it 2 ways, maybe more than this. The 2 ways that I know about are you can decide to give your own blood, spin it down, keep the red blood cells, right? Keep them in a fridge for a couple of weeks. Wait for yourself to replenish your red blood cell volume, right? And then shoot yourself up back with your own red blood cells. Yeah, you can do that. Or you can shoot yourself up with EPO. Either one will produce a competitive advantage in cardiovascular sports, but they can also do this, which is too many red blood cells, adding too much viscosity to the blood, causing you to have a heart attack or a stroke or a pulmonary embolism. And that has definitely occurred. Procrit is a drug. By the way, we have produced EPO in the lab now. We know how to make it. Procrit is one of the brand names. There's probably a whole bunch more for lab-produced erythropoietin. And they use it, for instance, if somebody is undergoing a cancer treatment, let's say leukemia. You have to kill off their bone marrow, right? And you do bone marrow donation, but part of the reason it's hard to recover is because you have reduced oxygen carrying capacity. So if I can shoot you up with Procrit, fake EPO, then you can increase your oxygen carrying capacity. So that's kind of what it's for, not just that situation. So it's been genetically engineered and now it's available, but because it's widely available, it's also abused in competitive sports. So what they do now, just like Talmadge, what they do now is they say, we're going to check your red blood cell level, yes, you competitive athlete, and I don't care how you got to 6 million. If you're above 6 million, you're not competing. So maybe you need to sit on the couch for 2 weeks if you're just a freak of nature, but whatever. And it's different. That's not necessarily that number. Go ahead.
Just as an aside, there's a cool documentary called Icarus about blood doping, specifically with cycling, but then they uncover like the Russian.
Ooh, I want to see that. I like documentaries. My kids are like, oh my God, another documentary. Shut up. Tell me that later. I'm going to forget it. I don't have anything to write down with. Okay, so destruction of red blood cells. What happens when they get old? When they get old, what happens is every single time they go through the spleen and the liver, like the big macrophages that are associated with the spleen and the liver, look at them to see if they look like they've got the crap beat out of them or if they have one more round left in them. When they start to look old and fragile and lose their shape, because the shape of a red blood cell is really important, then what happens is they get trapped by these macrophages in the liver and the spleen, and they undergo hemolysis, but they don't trash everything, they recycle. So they break down the globin to amino acids and dump it into the bloodstream. Yeah, because you can use amino acids, right? They break off the iron and send it into the bloodstream, and it goes back to the red bone marrow. You do lose some of it, but you don't lose all of it. Does that make sense? And then, so the iron and the amino acids are recycled. And a lot of the rest is waste products that you just didn't or couldn't capture. And it's eliminated as bilirubin that is made by the liver, goes into the gallbladder, is stored as a bile pigment. And that bilirubin is the reason your poop is that color. It's old red blood cells primarily. It's also the reason your pee is that color, unless you're taking multivitamins, and then it's going to be, Technicolor, right? Technicolor. Does that make sense? Your kids, you need to tell your kids that just to gross them out, okay? Tell them the reason their poop is that color. Unless you ate a whole bunch of Oreos, because we know what that will do to your poop, right? You don't? Experiment. Go do it. Okay. Clinical connections are anemia. I'm just going to tell you what it is, and then if we have time at the end, I'll go back to it. So anemia is just reduced oxygen carrying capacity. Before we get into the details, let's go ahead and say that anything that reduces your number of red blood cells, right, your shape of your hemoglobin so that it doesn't carry as well, or your amount of blood can cause anemia, right? It can be nutritional, it can be hemorrhagic, it could be a lack of vitamin B12, a lack of iron, right? Some hemolytic anemia, like when you get malaria, when they bust, all that kind of stuff. Actually, let's just talk about it now. I'll just do it quickly because I'm trying to get to clotting. Okay, I know that oxygen is used for cellular respiration, and if you don't, be very, very quiet right now because I will cry. Anemia symptoms, regardless of how you get anemia, the symptoms are fatigue, obviously, right? Shortness of breath. A lot of times you're kind of cold. Maeve, are you anemic? She says she's always cold. And you're pale because there's not that much oxygen bound to your hemoglobin. So here's some examples. Let's just talk about these really quickly. So after you lose blood or give blood, regardless of whether it was on purpose or on accident, you're going to have what's called hemorrhagic anemia. And that is a thing. I discovered that when I was in college. I think I was 19 or 18. And I was an English major, so please give me a break. I gave blood, and then I went and worked out. And that was not smart. And that's why everybody should take physiology in high school, because I slept for about 60 hours after that. Yes, and my roommate thought I was dead, but she hated me, so she didn't care. All my roommates have hated me unless I'm with them. Hemolytic anemia is when you have premature hemolysis of your red blood cells, like with a mismatched blood transfusion, right? Which is why when they first started doing blood transfusions, they were like, you're either going to get well or you're definitely going to die because it caused hemolysis. Or with malaria. Malaria is an intracellular red blood cell parasite. It's a protozoan. Just stick with me because this will matter in a minute. And it will infect your red blood cells, start going through its life cycle, and then cause lysis of the red blood cells. Does that make sense? Now, related to that and kind of interesting, guess where we used to get EPO before we could make it in the lab? It's not really a good human rights story. And guess what? A lot of times we are not the good guys in human rights stories. I don't know if you guys know that. So before we knew how to genetically engineer it, we used to buy the urine of malaria patients from other countries because they made so much erythropoietin because they had so much hemolysis that they peed half of it out. So we would buy the urine, we would extract it from the urine. I really, really hope that we also gave them anti-malarial medications, but I am a grown-up and I know better by now. So yes, that's where we used to get it. It's not a nice story. So that's hemolytic anemia. Aplastic anemia is when something happens to your bone marrow. Some drugs, some radiation, Chernobyl, that's going to cause you an aplastic anemia. All of the formed elements are decreased, both red blood cells, white blood cells, platelets. Therefore, the symptoms also include infections and bleeding and all that kind of stuff. So that's aplastic anemia. It's one of the reasons that, did you guys see the Chernobyl miniseries? So good. You should totally watch it. Wasn't it good? I was ignorant. I was severely ignorant. And then I listened to the whole podcast. You should totally watch it. Nutritional anemia is when you're missing one of the nutritional components to make the red blood cells. Insufficient amounts of iron, really, really common in women. Insufficient amounts of folic acid, vitamin B12, or protein. So a lot of times women who have this menstrual-related anemia, if they will front-load all of the nutrients necessary, they will have less rheumatic symptoms from the menstrual anemia. Renal anemia is when something's wrong with your kidneys. Does that make sense? Yeah? Kidneys need to be active in order to cause EPO secretion. So as you have kidney failure, it might seem initially counterintuitive that you're going to end up anemic, but you can and do end up anemic with kidney failure. Pernicious anemia, I love that name. Pernicious sounds like an old person word, and it's an old person disease. Pernicious. Pernicious. This is a lack of B12, but not due to a dietary problem with the amount of B12, but due to, as you age, for some reason, you stop producing this thing called intrinsic factor that's necessary for the gut to absorb B12. So dietary B12 is in meat, so it's not usually a problem in the United States anyway. B12 can't be absorbed without the help of intrinsic factor, and you stop making it as you age. So if you, for instance, are working in an elderly care home, what the hell do you call those? Nursing home. I was like old folks home. That's not right, Mel. Okay, do B12 pills help? No, you have to do B12 injections in that situations. Okay, and then last but not least, hereditary anemias. With hereditary anemia, it's actually kind of not an anemia, but it kind of is. It ends up causing anemia. Now they call it sickle cell disease. They used to call it sickle cell anemia. That was just one of the symptoms. That wasn't actually the cause. The anemia was just sort of the symptom. So what happens with sickle cell disease is it's a genetic disorder that changes a single amino acid in one of the chains, actually two, because I think it's a beta chain of hemoglobin. So it just changes one amino acid, okay? It's a point mutation in the DNA that changes one amino acid, causes a substitution. And what it does is it makes hemoglobin fold differently because you know the folding is related to which amino acids are in it. You with me? Okay. And when it folds differently, it actually sickles in low oxygen conditions. Okay. And that's really important because these barely fit through a capillary in their biconcave disc shape. And when they're this shape, do they actually fit through capillaries? No, they don't fit through capillaries. And when you go into a sickle cell crisis, which is a low oxygen causing sickling crisis, then what happens is tremendous amount of pain, but also you're cutting off the downstream capillary beds. If that happens, brain, heart, lungs, you have a stroke, right? You have a heart attack, you have some kind of pulmonary issue. So this was just a weird little genetic anomaly that occurred with an evolutionary selection component. And you guys have probably talked about this one before, but just briefly, I'll tell you about it. It's kind of interesting. It affects mostly people of African descent, but mostly Central Africa and then a few portions in Asia. It can affect any population because, of course, and any person because it's just a point mutation. But what happened was there was actually a genetic advantage, a carrier advantage in Africa. So here's what happened. So I can't draw Africa. A, I'm bad at drawing, and B, I'm bad at geography. But I think it looks kind of like this. That's not terrible. Okay, there's a middle portion. Shut up. It's a drumstick, and I like drumsticks. So Africa is not all a desert, right? It's a ginormous continent. It's got a whole bunch of different possibilities, and there are rainforests around the middle band of Africa, really, really moist. Moisture mosquitoes malaria, yeah? And so what ended up happening, historically speaking, is we had two selective pressures that were going on. And so this is an autosomal recessive disorder, sickle cell disease. And I'm not going to use S's because they're too hard to tell big from little. So I'm going to use A's. If I'm big A, I have normal red blood cells, which is basically from the side of biconcave disc, right? Biconcave disc. And if I'm big A, little a, normal with an asterisk, they look like a biconcave disc until you get into severely hypoxic situations, and then you might have a little sickling, but you might never have any sickling, okay? And then little a is sickle cell, and that is this shape, right? A sickle, right? Okay, so what happened is give yourself thousands of years and watch what happens to the population as you have two disproportionate causes of death, right? And sickle cell generally kills people before they hit their breeding years, right? So you have a disproportionate amount of death of people with sickle cell trait, yeah? Okay, but malaria doesn't like the sickled cells, okay? Does like the normal red blood cells, right? So you have a disproportionate amount, not all by any stretch of the imagination, of these guys who die of malaria, yeah? So who is more likely to survive? The one that's not sickled but not quite normal, right? Because it doesn't usually sickle, but the malaria pathogen does not like to infect them. So what you have is a carrier advantage. And so why do you still have so many people of African descent disproportionately carrying the sickle cell gene? Because selective pressure, right? Because of the evolutionary pressure for it. Does that make sense? There's kind of the same kind of thing going on with cystic fibrosis in Caucasian populations. The cystic fibrosis mutation provided some protection for childhood diarrheal diseases historically, so it's still really really common. Okay, so other anemias. There's the use it or lose it anemia, in which if you don't exercise, you don't need that many red blood cells, so when you try to exercise, you're not adequate to do so. And then there's hypothyroid-related anemia as well. Okay, so now we get into the stuff that is interesting and a little challenging, but not too bad. So let's talk about hemostasis and platelets. Okay, so you guys have known that platelets, also called thrombocytes, are responsible for blood clotting, yeah? But blood clotting is bigger than just clotting, so I want to use a different word. I want to use the word hemostasis, which implies, hemo implies blood, and stasis implies stabilizing, right? So this is the cessation of bleeding upon injury that includes clotting, but is not limited to clotting, okay? So when we look at hemostasis, there are multiple stages of hemostasis, okay? 2, there's 5. And most of them are pretty simple. There's only one that's actually complicated. So I'm just going to run through the big picture, the forest, and then we'll get back into the trees. So you don't have to rummage through your notes yet. So the first stage of hemostasis is going to be vasoconstriction. And this should make sense to you, okay? I have a vessel. I have a hole right here. And the blood is going this direction. What should I do here? I should definitely constrict, right? Because if the hose has a hole in it, right, kink the damn hose. Okay, so that's vasoconstriction. That's the first stage of hemostasis. That is involved in the cessation of bleeding, right? Just kinking the hose. The next stage you probably haven't thought about before. It's called platelet plug formation. It's when the platelets, not yet a clot, congregate around the site of injury. Okay, I'll tell you more about that in just a second. And then the third stage is clotting, also called coagulation. And we'll get back to that one. The fourth stage is clot retraction. And the fifth stage is dissolving the clot, fibrinolysis. Okay, and that's all in your notes, but just so you can see it up here as we're actually going through it. All right, so the only complicated stage is this one. The rest are relatively simple. Okay, so let's talk about them individually. Okay, so vasoconstriction, or the vascular spasm, is what happens immediately after you have damaged a vessel. So I want you to imagine that you've poked a hole in a vessel, either accidentally or you've just done an injection, right? And you poked a hole in the vessel. So what happens is in any vessel except a capillary, you can have vasoconstriction. Why can't you have vasoconstriction in capillaries? No smooth muscle, exactly. So they can't do the vascular spasms phase. So smooth muscle in the vessel wall constricts. It reduces blood flow. Cause, we put a question mark because we think that there's 2 or 3 possible causes, but we have yet to really empirically confirm how related they are to anything. But I'll go ahead and tell you that probably part of it is paracrine, yeah? In which it gets damaged. It says, hey, I'm damaged, and releases some paracrine agents that cause local constriction, right? And by the way, those paracrine agents, weird paracrine agents. ADP is released as a paracrine agent. There's something called thromboxane A2. Serotonin is released as a paracrine agent. But then in addition, they also think, I didn't write it here and I won't ask you about it, but they also think that the nervous system might help. Haven't confirmed it yet. Or it could just be the muscle's response to getting damaged, okay? Potentially all 3 of them, regardless of the mechanism. Damage, constriction, right? And the damage, the constriction is upstream of the damage. And then the next phase is going to be platelet plug formation. Okay, so I need to tell you what a platelet plug is and what it isn't, okay? So don't get these 2 portions confused. You don't have this until the 4th test, so you got plenty of time to think about it. But I want to show you what a clot is and then tell you that a platelet plug is not a clot. Here is a clot. Do you see all of these proteins that are involved? So a clot is really like a really, really sticky spider web that everything gets caught in that was in the area, okay? But this guy is a platelet, but that is not a platelet plug. A platelet plug is just when the platelets themselves actually congregate and maybe completely plug the hole, okay? It can and usually does lead to a clot, but if the hole is super tiny, it might be adequate without forming a clot, okay? So let's talk about how a platelet plug formation occurs. Okay, so this is kind of interesting to me. So if you remember a little bit of the anatomy of a blood vessel wall, you know, of course, that they have smooth muscle in the wall, anything but a capillary. But then they've got connective tissue right there, and then they've got epithelial tissue lining it, yeah? So does it make sense to you that in a healthy vessel, a platelet should never come into contact with collagen? Does that make sense to you? Because is collagen in epithelial tissues? No, it's not. So if a platelet comes into contact with collagen, it knows that damage has occurred. Yes? So what's going to occur is the formation of a platelet plug. So platelets contact the damaged vessel wall. It causes platelets to become sticky. Sticky is really polar. So a platelet can, for instance, end up having a whole bunch of positive ions and a whole bunch of negative ions. If you congregate the positives and negatives, then it actually becomes kind of electrically attractive to other things that are polar like that. So what happens is the platelet becomes sticky, and it causes sort of this platelet aggregation. And then as the platelet aggregation, they start to convene right near the hole. They start to release paracrine agents that cause the nearby platelets to be attracted and sticky. And the more platelets you have, the more paracrine agents you release. The more platelets you have, the more paracrine agents you release, until you're in a little positive feedback loop. And the positive feedback loop involves quite a few paracrine agents, but one of them is ADP, and one of them is that thromboxane A2. Anytime you see the prefix thrombo, it implies something is going to happen related to a clot. Okay, so platelets stick to one another. They stick to the damaged surface of the vessel. And there's actually another one called von Willebrand Factor that they're not showing here. And the only reason I'm going to tell you about this one is because it's got some historical significance. So von Willebrand Factor is one of the paracrine agents that starts this little positive feedback loop. It's secreted by platelets and by the damaged endothelial cell, and it binds to collagen, changes conformation, attracts other platelets. But von Willebrand Factor was involved in one of the historically significant forms of hemophilia called von Willebrand's hemophilia. And I would bet that Rebecca knows the historical significance of hemophilia in Europe. Do you?
Yeah, it's in the royal family a lot. It's in males, is when it comes down in the family tree, it exhibits in males. And that caused a lot of early deaths.
Yes.
Like 13, 15 years old.
Yeah, and if you combine that with the fact that they were trying to keep the royal bloodline relatively clean, then you ended up marrying your first or your second cousin. And yeah, and it gets passed on, and a whole bunch of the potential monarchs of Europe, or not necessarily all monarchs, but royalty of Europe ended up dying because of a combination of this X-linked recessive disorder and inbreeding. So it had some historical significance. I just picked on Rebecca because she has a history degree, and I was like, she looks like she knows. I'm not going to stress her out by asking. Okay. So the activated platelets, activated means when they become slightly polar. Yeah, they become like electrically attractive to other ones. They release other paracrine chemicals. You don't have to memorize all of these, but they release epinephrine as a paracrine chemical. They release serotonin, and that causes further vasoconstriction. So they sort of add to the vasoconstriction in addition to congregating. ADP, prostaglandins, thromboxane A2, which causes more platelet aggregation and more platelet aggregation and more platelet aggregation. It's a positive feedback loop. If the hole is really small, the platelet plug alone could stop bleeding. Yeah? If the hole isn't small, then that's just the beginning of what we are going to do. So we formed a platelet plug. This is why I always say that you don't want your platelets to congregate. Because sometimes when the platelets congregate, they spontaneously decide to form a clot, like teenagers and parties and pregnancies, right? Get enough teenagers around, what's going to happen? They're going to eat all your crap. If you got booze in the house, they're going to drink it, and somebody's going to get knocked up. Platelets, right? Okay? So that's why the basement has cameras in it. No, it doesn't at my house. I just tell them that it does. So I don't tell them. They wouldn't believe me anyway. My son's friends always say, is your mom ever serious? I'm like, you won't know when it's happening. Okay, so eventually the platelet plug could become big enough to plug the hole. This takes about 1 minute for the platelet plug to get about as big as it's going to get. But here's a question for you. Why does this platelet plug not just keep going and fill up the whole vessel, right? And it's kind of interesting, right? Because if you start positive feedback, why do you not just fill up the whole damn vessel? It is because these exposed collagen fibers are activating the platelets and causing this little positive feedback loop. But the other ones are going, move along. So the healthy tissue is releasing things that are saying, get off of my lawn, teenagers, get off, right? Okay, so it kind of puts a boundary on the size of the platelet plug. Does that make sense? So the unhealthy or damaged tissues are saying, come over here, and the healthy tissues are saying, get off of my lawn, right? I love this cartoon. Do you guys know the Awkward Yeti? He does science-based cartoons. No serious business. There's no time for shenanigans. Platelet party? Platelet party. Platelet party. Platelet party. Platelet party. So they're starting a platelet party. See, teenagers, get off of my lawn. Okay, so it doesn't extend beyond the site of injury because the healthy cells will release like nitric oxide as a paracrine agent, and they inhibit platelet plug formation. So platelet plug is not the same as a clot because it showed you what a clot was. But the more platelets you have, the more likely a clot is to form. Okay? Now, aspirin therapy. Aspirin therapy is prescribed for a lot of us with cardiovascular abnormalities, and it's relatively minor intervention that you can do. But we always talk about it as a blood thinner, but I just don't like that word because that's not what it does. It doesn't impact the viscosity of your blood to any great degree. What it does is aspirin therapy. So for instance, I've got atrial fibrillation, which means I'm more likely to form a clot inappropriately than you are. Aspirin therapy interferes with some of these prostaglandins that are involved in platelet plug formation. It is not a blood thinner. It's not even an anticoagulant because coagulation is when you form the clot. It's antiplatelet plug formation, which decreases the likelihood of coagulation. That's what that does. But you knew that, didn't you? Didn't you know that about aspirin? That it inhibits some of the things related to clotting, right? So that's what it does. So platelets are less sticky. They're less likely to catch and activate their friends. It prevents inappropriate platelet plugs and therefore inappropriate clots. But of course, it also prevents appropriate clots from occurring very quickly. So on the rare times when I am actually taking my daily prescribed aspirin dosage and I run into the coffee table, what happens? I get a ginormous bruise. Yes. Yeah. So huge bruising because it also prevents appropriate clotting. It takes longer for clotting to occur. So you bruise like crazy anytime you run into anything. Okay. It's not that big a deal, but okay. And now we get to the complicated part. And the complicated part is called clotting or coagulation. So listen to me for just a second. It's going to seem really overwhelming, and then it's going to clarify because it's going to get all messy, and then there's a summary at the end that will bring it back together. So try not to get frustrated. Okay, so clotting or coagulation is what we're going to talk about now. So generally speaking, the whole vasoconstriction is almost instantaneous. The platelet plug formation takes about a minute. Yeah? The clotting or coagulation, by the time you're finished with that, you're going to be 3 to 6 minutes. Okay? And then the other portion depends on how long it takes to heal. So what's going to happen is if you just imagine what blood is going to do if you let it sit still, it's going to clot. I don't know if you guys have ever seen this. Have you ever, it's kind of gross to talk about, but have you ever seen what blood does when it pools? It kind of squishes together and pushes yellow stuff out of the edges. Have you ever seen that? Right. The next time blood's just sitting somewhere, try to get yourself to not clean it up for a minute because it's interesting. So what happened, if you draw blood and just let it sit there, it's going to thicken and gel. And what actually happens is it forms a gel in the middle because clotting will occur in the middle, and then it pushes out whatever's not caught in the gel at the edges. That stuff is serum. Which is basically blood plasma minus the clotting proteins that stayed in there. So what do I mean by a gel? You know gelatin? Yeah? Gelatin is basically these long proteins, right? And they're from animal connective tissue. So gross when I think about it. Do you know how they make gelatin? They boil down animal connective tissue, so you have all of these collagen fibers that when you heat them up will go into solution, and then you stick a grape in it. It's so gross. When I told my vegetarian child about that, he's like, "They do what?" I was like, "It's okay, bro." Anyway, so what happens is you've got these proteins that under the right circumstances will get solidified, and they'll catch whatever is in the solution. So it's sort of this in between a liquid and a solid, because it's basically a solid with a whole bunch of liquids caught in it. So that's what basically a clot is. So when the gel shrinks, and for reasons we'll talk about in just a second, it will shrink, it squeezes out the fluid that's not caught in the gel, and that's the serum. You have experienced serum if you've ever had a surface blood clot, like a road rash, that when it shrinks up during the healing process, it squeezes out this clear yellow fluid. That's serum. If it's opaque, it's infection. But if it's clear, it's serum. Does that make sense? Okay, so clotting is a cascade reaction. It's like a domino effect, and it involves a whole bunch of different factors. They look super duper complicated, but they're not that bad. Hold on, let me pull it up. I didn't pull up the right one. 15:8, 15:9. Just wait right there. Okay. I do simplify it for you quite a bit, so it's not as complicated as it originally looks. So here's clotting. You don't have to memorize all of it, though, so don't walk out, okay? Here's clotting. Okay, but before you get to the details, I want you to look at the big picture. What is going to happen ultimately is you get this stuff. Fibrin is the fibers that you saw in the picture. They will form the gel, right? They're actually going to form the gelatin that's in there, okay? So that's the point. And then during the process of getting to fibrin, what you're going to have is a cascade of one thing gets activated, activates another thing. One thing gets activated, activates another thing. So just look at that really quickly. This one gets activated, activates that one. This one gets activated, activates that one. This one gets activated, activates that one, all the way down. So it's called the coagulation or clotting cascade, okay? You don't have to memorize all of it. You're going to memorize the important points of it, like the beginnings, the middle, and the end, okay? All right. So the coagulation cascade, it's a domino effect involving a whole bunch of different factors, most of them proteins that were sitting around in the blood all the time just in case you needed them. Does that make sense to you? They're sitting around inactive. It's not as if I have to make them when I need the clot. They're sitting around in an inactive form. Why not just make them when you need the clot? Yeah, because time constraints. Time is of the essence when you have a hole in your blood vessel. So what I need to do is I need to have them sitting around. Most of them are made by the liver, okay? And they're just sitting around waiting to be activated. So we'll talk about the activation mechanism and what happens at the end as well. So clotting usually takes 3 to 6 minutes. And you've got this gel, which is formed by these protein fibers called fibrin. That's where we're trying to get. It's a network of insoluble fibers. They're not soluble in blood plasma until you activate an enzyme which will dissolve them. And it's going to be this domino effect. At least 13 different factors are involved in coagulation, but it's not as complicated as it originally looks for 2 reasons. First of all, I don't make you memorize the whole thing. And second, there are fewer than you imagined because 12A is just activated 12, okay? 11A is just activated 11. 9A is just activated 9. Why the hell are they in this order? They're in the order that people discovered them, not in the order that they work. I apologize, okay? But it's okay. For me, I don't make you memorize the numbers anyway. You can do that in patho if you need to. Do they make you in patho? Anybody taken patho? Oh, you guys haven't taken patho yet. I'll ask my students who have taken patho. So there's at least 13 different factors, but the activated one just has an A on it, okay? Now, the other thing that is important about clotting before we get into the detail, just to look at the forest instead of the trees, is how important calcium is in clotting. I've told you guys about 10 times this semester that calcium is necessary for blood clotting, right? But now I want you to look at where calcium actually is necessary for blood clotting. Here's calcium, here's calcium, here's calcium, here's calcium. You got a calcium problem, you got a clotting problem. Yeah? Too little calcium, you are not going to clot effectively. Too much calcium, you might clot when you didn't mean to. Does that make sense to you? So calcium is really, really important in clotting, which is why PTH and calcitonin are directly related to sort of blood clotting disorders in a way that initially seems counterintuitive, but now you're like, okay, that makes sense. Okay, so the other things that are necessary for clotting before we get into the details are vitamin K. Hardly anybody ever talks about vitamin K, unless, of course, you work in labor and delivery, and then you do talk about vitamin K. Okay? Vitamin K is a lipid-soluble vitamin, And what is necessary to do is to make this guy right here, okay? Notice that without this guy right here, you are not going to get to the end regardless of how far you came, right? So vitamin K is necessary for the liver to make what we call prothrombin and also some other factors. Vitamin K, this is important to talk about because some whack stuff is going on with vitamin K lately. Vitamin K is made by your normal flora, primarily in your large intestine. Your bacteria make it for you, so stick with me for just a second. It's lipid soluble, which means that if you got a bile problem, you're going to have a vitamin K problem, liver problem, vitamin K problem. And then the other thing is that you don't make vitamin K before your intestine is colonized with your normal flora. So when you are first born, right, you don't have your normal flora. You're not like completely sterile because you got some come from your mom. You actually, if you're delivered vaginally, you're more likely to get your vitamin K population a little more quickly. But primarily what you have to do is you have to eat. Yeah, you have to colonize your intestine with vitamin K. And it usually takes around 8 days after birth for your bacteria and vitamin K to be adequate for blood clotting. Okay, so this is really important for us to understand. Because what they do is, they didn't used to give you the choice, and now they do. And I'm not sure this is a thing that people can be trusted with at this point. When the baby is born, because birth is a little rude, and sometimes they have to poke babies, and sometimes crap happens to babies, they are really, really high risk if they get injured of bleeding out because of the lack of vitamin K. Does that make sense to you? So what they do, and they have done for decades, is give them a vitamin K injection to help them for the first 8 days until it's colonized. It's just a little protective mechanism. But anybody work in labor and delivery? No? Sometimes people do. But what has happened, roped into this whole anti-vax thing, oh yeah, it's totally happening. Babies are dying because people have a tiny bit of education from Google University and they think that anything that comes in a syringe is a vaccine. And so in addition to being anti-vax, they are also anti-injection for their babies. And their babies don't get a vitamin K injection. And we hope that the baby is not going to get poked or damaged. But if you have that combination of the baby who has no vitamin K injection and needs an intervention of some sort, then you can have babies hemorrhaging and bleeding out when that was completely preventable, which is just mind-blowing. But if you have any friends who work in labor and delivery, ask them. It's totally a thing. It has happened in Utah. I think the last time I looked, it's not dramatic because you have to have sort of this Venn diagram of things that happen, right? You've got to have somebody who doesn't get the injection and then some kind of injury that occurred. But the last time I looked, 2 babies over the last 3 years have died due to lack of vitamin K injections. Completely preventable. But of course, once the hemorrhage occurs, the vitamin K injection is not adequate to produce prothrombin quickly enough to actually cause the clotting. Does that make sense? So it's a protective mechanism. Does that make sense? So anyway, this is really interesting. So they do vitamin K injections, and they've been working for ages and ages. So I already talked about this, but each clotting factor basically has a Roman numeral associated with it, and then when it's activated, it's going to get an A. Everybody good? Okay. So let's talk about the 3 stages in the formation of a clot. We'll do the easy parts first. Okay. So the first stage is getting to prothrombinase. This enzyme right here, factor 10A, right here, is prothrombinase. And I think I put this. Didn't I put it in your? Oh, I didn't. In your, I'll post it for you. I've got a really good diagram for you guys. Prothrombinase. So kind of the first thing is getting to prothrombinase. I lost my place. Okay. So however I get here, factor 10A is prothrombinase. Okay. And there's 2 different ways of getting there, extrinsic and intrinsic pathway. Okay. But then after I've got prothrombinase, which is factor 10A, I have to go to the next stage. Look at what prothrombinase does. I'm going to draw it on the board for you in just a second, but we're going to look at it in this figure first. Prothrombin Ease is going to convert prothrombin, pro means before, to thrombin, yes? Okay. And thrombin is going to convert fibrinogen to fibrin that gets stabilized and forms the clot. So let's look at what happens here. This is not terrible. It just looks terrible. Okay. So prothrombin Ease converts prothrombin, as in pre-thrombin, to thrombin. Thrombin converts fibrinogen, as in the genesis of the protein fibrin, to fibrin. And fibrin forms the clot. You have to stabilize it, but I don't hold you guys responsible for the stabilization mechanism. Does that make sense? Okay. So let's talk about where these things actually come from. Okay. So the thing, we're going to come back to prothrombinase in just a second and have to how you get there. But once you've got prothrombinase, which is 10A, by the way, factor 10A. Once you've got it, it converts prothrombin to thrombin, and thrombin converts fibrinogen to fibrin, which gets stabilized, and all of these little fibers that you are seeing are fibrin. Okay, think of them as really, really sticky spider webs. Okay, so let's talk about how that occurs. Fibrin is stabilized by fibrin stabilizing factor. So now what we need to do is we need to talk about this part, which is how you get here. There are two different pathways to get there. There is the extrinsic pathway, or the intrinsic pathway. And it kind of depends on whether the blood starts the process or the damaged vessel, damaged tissue starts the process. Which one do you think is which? Blood starts the process via what pathway? Intrinsic. Damaged vessel, extrinsic. Good. And sometimes they're working together at the same time. Okay, so let's look at what's going on with that. All right, so when you get here, by the way, this is called the common pathway because it doesn't matter how you get there. It starts about prothrombinase. Okay, so the common pathway begins when you get prothrombinase. Regardless of how it was initiated, extrinsic or intrinsic pathway. So let's look at what happens. So let me tell you about the extrinsic and the intrinsic pathway. The extrinsic pathway, as you guys already figured out, of forming prothrombinase is initiated by chemicals that are released from either the vessel or the damaged tissue. Okay? The damaged tissue will release something called tissue factor 3 or TF3. So listen, it'll get simpler. TF3. The damaged tissues around or the vessel release tissue factor 3 into the bloodstream. It's paracrine agent. And then what it will do is activate a complex cascade that gets you here. I'm not going to hold you responsible for all the intermediate portions. Okay, we will look at it in just a second though. The intrinsic pathway is released from those sticky platelets, the platelets that congregated. So, and what it's going to do is not release tissue factor 3, but platelet factor 3. I don't know why that one uses an Arabic numeral and that one uses a Roman numeral, but it just does. So, the sticky platelet that were formed when they started to congregate will release PF3, and that's going to get me to the same place. Okay, go ahead.
Sorry, PF3 should be in.
Oh, sorry, did I switch them? Thank you. I wasn't trying to confuse you. TF3. Thank you for catching that. Okay, so let's just look, and then we'll clarify. It's going to be okay. Okay, look. What happens when platelets come into contact with collagen is they get sticky, right? Ultimately, that's going to cause them to release platelet factor 3. This is the intrinsic pathway. to get me to prothrombinase, factor 10A. Good? In the extrinsic pathway, what happens is ultimately I release tissue factor TF, or tissue factor 3, which gets me ultimately to prothrombinase. It's two different ways of getting to the same place. Everybody okay with that? Okay, so regardless of how I got there, when I get to the common pathway, I'm going to get a clot. So before we do a summary and clarify it, so what would happen if I damaged the vessel wall and had a platelet plug, extrinsic or intrinsic? Both. Yeah? And they would be amplifying and making the response faster. But what happens in a person like me who has platelets congregate in her atria because of atrial fibrillation? Which one is the culprit? Intrinsic pathway, yes? The intrinsic pathway started it without an actual damage. That's why you want to watch your teenagers, right? They're starting a clot and there wasn't a hole. That's because that's what they start to do. They start a platelet party when enough of them congregate. Okay, so let's just do the summary and then we'll be okay. So the summary is this. So let me tell you how to read it and then it'll be fine. And I'll post a figure for you too. I know you won't look at it until after your test, but here's the summary. So the extrinsic pathway, I want to tab that over a little bit. The extrinsic pathway is activated by tissue damage. I'm going to read down though, which releases tissue factor 3, yes? Which activates prothrombinase. That's where the common pathway starts. Prothrombinase converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. Fibrin is stabilized to form a clot. Yes? Isn't that what the extrinsic pathway does? And the intrinsic pathway, let's read it. The intrinsic pathway is activated by an injured vessel with exposed collagen that causes sticky platelets. Yeah? Although the platelets will do it without the collagen if they congregate for long enough, FYI. Okay? Causes sticky platelets, which releases platelet factor 3. Yes? Which activates prothrombinase, which converts prothrombin to thrombin, which converts fibrinogen to fibrin, and fibrin is stabilized to form a clot. Yeah? I mean, not do you like it, but is it less complicated than it looks in this picture? Yes, it is less complicated than it looks in that picture. Everybody okay? Okay. The last two phases, and then we'll do clotting considerations after your test. We can do these last two phases because they're actually really easy. The last two phases are clot retraction and fibrinolysis. Clot retraction is the tightening or contracting of the fibrin clot, which tends to pull the edges of the damage together. And they figured out recently that that is because platelets have actin and myosin in them. Weird as hell, right? And whenever it's almost done, the actin and myosin will start this teeny tiny little cross-bridge cycling, and it actually pulls the sides together, and that's when you squeeze out the serum. Yeah, pulls it together. And you guys have felt that, right? When you feel a clot or it feels itchy and pulls us together and gets really tight. And then the last phase is just the fibrinolysis phase. And this is easy. So what was the clot made of? What's the protein? Fibrin, yes? Okay, here's the deal. When you are done, the healthy tissue starts releasing different paracrine agents that says, I'm cool, I'm fine. Yeah? And the damaged tissue releases something called TPA, which is tissue plasminogen activator, as it heals. And TPA converts plasminogen to plasmin, and plasmin dissolves the clot. So where did TPA come from? Healthy tissues. Healed tissues release TPA, tissue plasminogen activator, and they convert plasminogen to plasmin, and plasmin dissolves fibrin. Okay, and the last little tidbit before you guys leave is just that TPA has now been genetically engineered. We can make it in the lab. It is fantastically effective as a clot buster in someone, for instance, who had a massive stroke and you don't want them to have another one, but you will bleed out like a sieve if they can't undo it really quickly. So this is not a maintenance medication. This is a medication that they give inpatient. It's also outrageously expensive. So unless you have really good insurance, you probably won't get it at all. Okay, so Tuesday you will have your test. I'll post a figure for you that kind of clarifies this. I meant to put it in your notes. I don't know why I didn't do that. And this is not on your test. For what? I don't know how your brain works. I am a reading writing learner. I rewrite everything over and over and over again and draw flowcharts. Summary pages, lots of flowcharts. Make your own concept map if you can, right? Either on paper. No, you could do it on paper too. Yeah. Okay, so